Hello, my name is Juanita Bailey, and my research involves pesticide degradation through Burkholderia. Burkholderia is a free-living bacteria that resides in the soil and is capable of participating in symbiotic relationships with many insect hosts. Now, the research we chose this as part of our study is because we're seeing an increase in the global population, and this population is projected to increase by 12.5 billion over the next 20 years. This also means there's an increase in agricultural demand and a rise in chemical pesticide use. Now, this poses a problem in agriculture when there's this excessive application of a single pesticide that causes accumulation in the soil, creating an ideal environment for the development of pesticide degrading capabilities in these free living bacteria, such as Burkholderia. Now, our isolate of interest is Burkholderia, which is known to form and establish a symbiotic relationship with Leptoglossus, the leaf-footed bug. Leptoglossus is a crop pest that typically affects citrus plants and causes tons of damage. And for our study, we isolated 16 potential pesticide degraders from across all over the United States, such as in field sites such as Arizona, North Carolina, and even Texas. And to determine whether our potential degraders can degrade pesticide, we perform a degradation assay where we expose the Burkholderia live cell cultures to pesticide, and then we place them in a shaker for incubation for 72 hours at 25 degrees Celsius. Now, it's important that this shaker is closed and sealed from light to ensure that there's no pesticide degradation through light exposure. And but after this incubation period, we separated the live cell culture from the pesticide, and we did that by performing a chemical extraction. And after the chemical extraction, we dried the pesticide to prepare it for HPLC analysis, where we found that we have one potential degrader, and that is a degrader of an organophosphate, phenyltrethione. And we found that this isolate is one that is found in Leptoglossus philopus, which is one of the leaf-footed bugs that is native to Texas. Now, the HPLC analysis shows that there's a 26% reduction in the phenyltrethione content from the phenyltrethione live cell culture when compared to the pesticide blank and normalized using an internal standard of EPN. And this is especially important because we've established that Burkholderia are capable of forming symbiotic relationships. And if they have these degrading capabilities for pesticide, then they're able to pass on this pesticide resistance to their hosts, like Leptoglossus. And this is especially important because the costs associated with pesticide resistance in agriculture are upwards of 1.5 billion per year. And this means that there needs to be new methods for circumventing these kinds of problems, such as a rotational schedule with pesticide. And this is to ensure that not one single pesticide accumulates in the soil, which would create the optimal environment for these free-living bacteria to develop these pesticide degrading capabilities. There is more to be learned, though, such as the metabolic pathways that are involved in this pesticide degrading traits in Burkholderia. And we have other projects in the future where we can visualize the point mutations of Burkholderia as they are exposed to higher and higher concentrations of pesticide. And this is through the construction of a microbial evolution and growth arena, otherwise known as a megaplate. And with this, we can visualize these point mutations and see where the Burkholderia overcome these higher and higher concentrations. And we're very excited for our next projects and the results from that.